Welcome to the Mind and Matter podcast. I'm your host, Nick Jackomis, and today I'm speaking with Dr. Reggie Gaudino. Reggie has a PhD in molecular genetics, and he's been working for a number of years in the biotech and cannabis industry, broadly speaking. Reggie worked for a number of years at Steep Hill Labs, one of the first and one of the largest cannabis testing laboratories in the legal cannabis industry. And Reggie is an expert in the biochemistry and phytochemistry, as well as the growth properties of commercial cannabis, also known as marijuana. He is currently the Vice President of Research and Development at Frontrange Biosciences, where he leads a team of scientists, including agronomists, breeders, and growers that carry out sophisticated breeding programs for hemp and cannabis. Reggie and I talked about a variety of topics related to the biochemistry and phytochemistry of commercial cannabis, including what the history of breeding has looked like in the United States, what the characteristics of this plant are, what its chemistry and biochemistry and its genome actually look like, and how those things interact to determine the final chemical profile that any given plant will express. And we also explored the relationship between the various nomenclatures and labeling system used in the consumer cannabis industry and the actual scientific understanding we have of the underlying chemistry. So, for example, people often in the industry refer to Indicas and Sativas, Indica strains being those that are allegedly sedating and Sativa strains being those that are allegedly energizing. We talked about whether or not the categorizations as they're used in the industry actually make sense and actually map onto the underlying chemistry of what these plants actually look like under the hood, so to speak. So if you're interested in cannabis or phytochemistry, this will be an interesting episode. We dig fairly deep into this topic, so Reggie goes into a fair amount of detail. But if you're interested in this topic, this is definitely a good one. As always, if you enjoy the content on this podcast, please like, share, or subscribe. You can subscribe to my weekly newsletter at mindandmatter.substack.com. You can subscribe and watch video versions of these episodes on YouTube. Today's show is brought to you in part by Dosist, an all-natural cannabis company specializing in dose-controlled cannabis products made with plant-based ingredients. To learn more about Dosist, their products, and where they are available, please visit their website through the link in the episode description. This episode is supported in part by Athletic Greens. Their main product, AG1, is a comprehensive and convenient daily nutrition product containing 75 vitamins, minerals, and whole food-sourced ingredients with less than 1 gram of sugar per serving, no nasty chemicals or artificial anything. It's gluten and dairy-free and compatible with paleo, vegan, vegetarian, and ketogenic diets. AG1 is a quick and convenient way to supplement your diet to help ensure your body is getting the nutrients it needs. It comes in powder form, and you can mix it in water and drink it, or you can put it into a smoothie or a shake or something like that. I mix it into water and drink it with the first meal of each day, and it's super convenient. If you go to athleticgreens.com/mindandmatter, Athletic Greens will give you a free 1-year supply of vitamin D and 5 free travel packs with your first purchase. Their vitamin D product comes in tincture form, so you just take 1 drop each day. A large fraction of the population is actually vitamin D deficient, especially in winter months when we get less sun exposure, and vitamin D is super important for the proper function of the immune system and for a variety of other things. And there's even evidence indicating that vitamin D deficiency is correlated with more severe cases of COVID-19 in those who get infected. Every time I go into the doctor each year for a checkup, I'm always told that vitamin D deficiency is very common and I should be supplementing on a daily basis. So visit athleticgreens.com/mindandmatter or click the link in the episode description. You'll get a free 1-year supply of vitamin D with your first purchase. And with that, here's my conversation with Dr. Reggie Gaudino. Reggie Gaudino, thank you for joining me.
Hi, Nick. How are you? Thanks for having me.
Good. Can you give everyone just a short background on who you are and what your scientific training is?
Okay. My name is Reggie Gaudino. I'm the VP of R&D at Frontrange Biosciences. I previously was president and chief science officer at Steep Hill Laboratories, which was at one time the largest and was, I believe, the first commercial cannabis testing lab in the world, but definitely in the United States. My background is in molecular genetics and biochemistry. Got my PhD at Roswell Park Cancer Institute, which is part of the SUNY system. Did my postdoc at Washington University in St. Louis, where I worked on went from bacteria and yeast into plants. That's where I started getting my real plant training. I worked on a project that actually involved using ethylene gassing chambers that were supplied by Monsanto. So I've actually been inside of Monsanto's headquarters, or at least the research facility in Chesterfield, for quite some time, good part of my graduate career. Then got into intellectual property and made noise in the beginning of my career in the cannabis industry about intellectual property and the importance of it. And kind of my path to this point.
So you're basically a molecular geneticist in terms of your scientific training, and you've been working in the legal cannabis industry for some time, mostly in the lab testing and science space, let's say.
Right. So and went from developing through the work at Steep Hill an understanding of the basic chemistry in a very deep way, and a way I think that other people had not gotten to at the point. And then from there was able to, because of my background in molecular genetics and biochemistry, and genomics, right? So while my background was in molecular genetics and biochemistry, so I'm old enough to have started at the beginning of recombinant DNA, right? So I've done every type of sequencing. Bioinformatics is a term that did not exist when I got my PhD, and at the time we did it by writing out sequence on graph paper and moving pieces of graph paper against each other to do a lens, literally, right? So I've done every aspect of molecular biology and genomic kind of sequencing study. Then went because of my intellectual property training, which was at Sequenome, which helped invent the first non-invasive pregnancy test through the doing a fetal karyotype through the blood draw from the blood draw of a mother and looking at apoptotic DNA. It developed, when we were there, we developed a lot of IP on sequencing and karyotyping and how to use that information in building, genome research programs, right? So when I left there and came into this industry as going back into science from intellectual property, a lot of that genomics came along with me and formed the basis of what we did at Steep Hill. At Steep Hill, we introduced the first sex test, the first CBD test. Unfortunately, it was not popularized and sold as well as it should have been. Steep Hill really was at the forefront of a lot of stuff in the industry in terms of R&D. And then from building those tools, we went in-house and are now breeding. So my entire team is focused on breeding and actually actualizing some of the stuff we learned, right? And a lot of this talk later will be about terpenes, and that's really where the nitty-gritty lies. And we've focused very heavily on the terpenes. Published in 2019 in PLOS One, that the first complete description of the terpene synthase family in cannabis, and then have been going from there. So, and just recently, ironically, something that we left behind at Steep Hill finally published. It was a patent application on chemical profiling and using that chemical profile as a prediction tool on the effects of potential various chemovars. So, and then I know you and I talked about that a couple of years ago.
So we're going to go fairly deep into some of this stuff. In order to give people who are less familiar a foundation, I want to ask some basic high-level questions about cannabis and just have you kind of set that foundation for us. So could you maybe give a concise summary of what kind of plant cannabis is, where it first evolved, and what kind of environments it was native to before it was domesticated?
So that's a great question. And I'm not sure anybody can actually answer that question. It's been around so long and it's been domesticated for so long. The idea of land races, I think, is a misnomer, right? Because even the land races as we know them, right, quote unquote, seem to have a high degree of hybridization. And the fact that we did in fact now find, cannabis from almost 2,500, 3,000 years ago that was high THC varieties, right? So so even back then, there was evidence that there was some separation of cannabis into its, separate types, the type 1s, the type 2s, type 3s, going back a long time, right? So we do know that some of the basic types are you have the Afghani types, the ones that look more like the Indicas, and so they range from the Pakistan, Afghanistan, Himalayas, that area there, right? Then we have the Siberian types, right, which is the ruderalis, right, which is a completely different looking thing, but is where where lore says we got a large portion of our autoflower from. And then you have the more equatorial types, the ones that we typically call the sativa types, right? Where you have, they're more, they have evolved for their particular environment, right? So you had the ones that evolved for short day, colder environments, short growing periods. You had ones that essentially grew all year round and languished and produced flower at, because of a different set of chemicals. They're less photoperiod sensitive, or actually rather they're not that they're photoperiod insensitive. It's just that there's additional triggers for their flowering program, right? So because they exist in 12-12, right? So it can't be light, right? So, and I think when you look at those basic types, traditionally or historically, right, those forms did in fact seem to travel with a function, right? So the long, lanky sativas seem to have their chemical groupings, and those chemical groupings likely came from the environments that they were exposed to, right? So now the question going back to, the domestication, right, and what effect that has had on it, right? So one thing to understand is that built into the DNA of cannabis is this tremendous plasticity, right? One way to look at cannabis is it is in fact a early colonizer species. You find cannabis is able to adapt its growing regime to more environments than most other plants, right? So, and it's very human-like in that response, right? Like, I mean, humans can go almost anywhere, right? We live everywhere except, Antarctica, right? And the Arctic Circle, right? And when we try hard enough, we can even live there, right? But you find cannabis living in all the same environments that you can find humans living in without extreme adaptations, right? And so because of that plasticity, right, and because its seeds were nutritious from the beginning, right? So early hunter-gatherers, they ate seeds, right? That, they ate, and so we found that we could in fact subsist on some of those things. And the fiber ended up being useful for clothes and for rope and for other things. And so it became rapidly a human survival toolkit kind of addition, right? So, and that occurred, early on. And so I don't think you can talk about human of evolution without talking about cannabis as part of that, right? So it is in fact one of the earliest, if not the first plant to be domesticated. There's evidence that it was domesticated before things like wheat or whatever, right? So, so I think one way to look at why we should care about what cannabis does is because it is in fact so adaptable, right? And that means that it must have systems in it that allow it to rapidly change. If you look at cannabis and it comes from, I think so it's no longer a rosacea. I think hemp and cannabis have been moved. I think it just happened in the last month or two. They moved from the rosacea family to something else, but I forget that. So, but it was just done, reclassified, right? So, but if you look at where it was before, and even its closest relative, hops, right? Those are not, annuals. Those are perennials, right? Those are plants that live and thrive and come back year after year after year, right? And so the question that I've always had about cannabis is it doing what it's doing because we forced it to? right? Like, is it a plant in transition from being a perennial to an annual because of the way that we forced it? If you look at the equatorial species that grow in Jamaica and other places, right, they flower year round. They just go and they hack off pieces and they leave the plant right. So this has always been a fascinating thing to me, right? So what is cannabis, and are we seeing the effect of a lot of human intervention on its growth program, right? Or did it really evolve completely different growing programs because it grew at the equator, and then it was also growing up in, on the mountains in the Himalayas, right? So fascinating things to talk about.
Yeah. So cannabis is a very adaptable, malleable plant, which is very interesting. So it can take many physical forms. Its genetics are very interesting. The chemical diversity, which we'll talk about, is very interesting. Sticking on the physical side for now, especially for people who aren't that familiar. So when you buy cannabis flower, when you see a bag of weed, whether it's in the illicit market or you go buy some at the legal dispensary today, people call that the flower, or they call it the bud. What part of the plant is that, and what is that part of the plant actually doing for it?
So that is in fact so cannabis is actually the bud is a fruit, right? So we call it the inflorescence, but it's typically not, because flowers don't make seed, right? Well, a specific part of the flower makes seed, right? So we're looking at a highly elongated, ovary, right? Like, I mean, so it's So it is in fact the inflorescence, right? It is the flower, right? But that flower turns into the fruit, because that's the fruit is where you get your seed, right? So the bud ends up becoming, I think there's a term for that.
But it's the female reproductive organ.
Right, exactly. Right. But and so whether it's a flower or it's actually the ovary, fruit bearing, part of the plant, is a good question. But it is technically, when we think of flower and we look at a rose, right? There's a difference. So that rose, produces pollen, or it will then produce a seed in a very specific part of the bud. Whereas the flower that we smoke is in fact, just there to produce seed, right? So, and we've cut that off by growing sensimilia, right? So, but what's interesting about it is that flower is a highly specialized organ, because if you look at male flower, you don't get, terpenes, and you don't get all the THC produced. And so there's structural differences between the male and the female parts of this plant, that are highly important and play into this whole thing we're about to talk about. So
interesting. And then maybe the last piece here is, so if you zoom in and look at these buds or these female flowers very closely, there's these small structures called trichomes, which are going to be very important for our discussion. So what exactly are those, and why are they important?
So trichomes are an interesting thing. So if you look at the plants that make trichomes, typically trichomes are on the outside of a plant to do one thing: to house things that are bad for the plant but good for survival, right? So generally you find trichomes on the outside of the plant, and there's several forms of trichomes, right? So we look at the glandular trichomes as the important trichomes, because that's where all of the chemistry happens for the plant. But there's like 3 or 4 other types of trichomes, some of which are actually weapons or weaponized, right? And so a lot of the, a good example of this is creosote, right? So creosote puts the toxic chemical, right, on the outside of the plant in its own trichomes, right? And the trichomes are pointy and sharp, and cannabis has tiny little pointy sharp, trichomes on the underside of the leaves and the stems as well, right? So and so trichomes generally are a protective adaptation for a plant, so it can help ward off things that would either predate it, like herbivores or whatever, or fungus that would inhabit it, right? So or infect it, right? So fungus or viruses. So generally, trichomes are in fact an adaptation for the plant to survive, right? But so it can put out toxic compounds that won't affect its own growth.
Interesting. So the trichomes are the structures that contain a lot of the interesting chemistry. A lot of this chemistry is actually bad for the plant, which is why it's on the outside, and it's really engaging in chemical warfare, I guess.
Yep, exactly, right. And that's an important thing to understand, right, as we move forward through this conversation, because we look at terpenes and the chemistry of the plant as beneficial to us, right? Mm-hmm. And sure, it is totally by happenstance, right? So it was a total mistake, because these things are actually produced not for us, but for the plant to survive, and it's surviving against insects that want to eat it, and fungus that wants to turn it into mush, right? I mean, so and these chemicals are there to help its own survival, and the way it does it through the biochemistry and the systems that it plays into, right? It just so happens that in our evolution, some of these systems ended up being critical functions for higher cognitive thought or whatever, right? So it's interesting.
So we've already mentioned that cannabis is very plastic. It's very adaptable, and there's many different forms it can take. So how do you start to think about the different types of cannabis that are out there, and what actually defines, what's one type versus another? And I guess this could be a part of the conversation where we differentiate between some different terminology that you often hear. So a lot of people talk about different cannabis strains. You also hear people use the term cultivar, and then you also hear other people use the term chemovar. So what do these terms mean, and how do you personally think about what truly differentiates distinct varieties of cannabis?
So strains is scientifically typically used for microorganisms or viruses, right? So serotypes, strains, that kind of thing. So I like the term cultivar. It denotes much the same thing, and because these are cultivars, so you're taking a genetic population, and you are going after particular phenotypic characteristics, and or some of it which is chemical output, right? And so you then select from a genetic population, right? And so these become your cultivars, right? So now. Or varieties. Cultivars and varieties are interchangeable, right? So now, the problem with cannabis is that any given genetic, because of the plasticity, right, can in fact, under different conditions, give you different chemical output, right? So a cultivar could theoretically give you different chemovars, right? Because of, again, the plasticity and how the plant will respond, right? So, and some of that is inherent, right? Like, so, even if you have inbred lines that you understand exactly what terpene synthases are being carried, right? So they don't all fire at every given time, right? Because they are in fact environmentally responsive. So the same genetics, even though it looks identical in the field, if you don't lock that growing environment in as well, you may in fact get a different chemical output. Will that chemical output be vastly different? Probably not, but different enough that it could cause, if it's being used for medicine, it could cause a different reaction, right? So some of these things are important, and it is important to understand the differences, right? So, you have cultivars or varieties, and cultivars or varieties can in fact give you different chemovars, right? And strain can be used, but strain is typically a microorganism term. So
I see. So cultivar basically just refers to different types of plants defined by their phenotypic features, the way they look, or their growth patterns, or their chemistry. And strain is sort of it's an informal term that the cannabis industry has used, even though it's not really what a scientist would use for a plant.
Well, I mean, even in agriculture and plants, right? So in most agriculture, even they're called cultivars, or they're called in the ornamentals in particular, right? So, and ironically, because ornamentals are largely clonally propagated, much like cannabis, right? So there is a direct kind of like correspondence between agricultural speak and cannabis speak, right? Like we're doing the same things, right? So clonally propagating specific genetics, right? So because it's a specific genetic, right, it is in fact a cultivar, right? Or a, it would, and the phenotype, and so that's another important thing that people need to understand, right? So phenotype is actually a biological equation, right? Phenotype is equal to genetics plus environment, right? So phenotype can only be 100% the same if the genotype and the environment are 100% the same all the time, right? As soon as you vary one, this thing changes, right? It's a biological equation. It's just like math, right? So and that gets into the intellectual property thing, right? Like, people want to have like these broad patent claims on a cultivar, right? But like you can't, because a cultivar could be different chemo profiles and you could step on everybody else's stuff. So in my opinion, if you want a really solid patent, or the patent office should require a tie of genetics to cultivation methodology to give you your chemotypic, output, right? So there's a whole bunch of things that cannabis does that steps across so many lines and makes it so complicated. It's really like internally for us, it's been like, cause cannabis, right? So like that's our internal meme in my R&D team, right? Like it's because nothing happens the way you would expect it, or it should, because it's cannabis.
So because the plant is so adaptable, you can have two different plants with the same genome, but they actually express that genome in different ways depending on the environment they're being grown in. I mean, it sounded like what you were saying is, if these plants are using all of these chemicals for defensive or protective purposes, there's genetic programs that allow the plant to shift, say, the terpene it's making if some type of mold comes and infects it. Is that the basic idea?
Yeah. So the plant is full of its own sensors, right? And actually terpenes are part of that response system. So a lot of monoterpenes, right, are actually what are called herbivore-induced plant volatiles, right? So monoterpenes are the ones that alpha pinene, the ones that are the first to go away when in a poor cure, right, kind of thing, right? So a lot of these things are designed to be very spreadable, right? Like you find monoterpenes in the soil because they volatilize and they can travel. It's there for either chemical defense or adaptation, right? So some of these terpenes are repellents. Some of these things are attractants. So some of them will attract a predator on something that's eating you, right? So kind of thing. So it's a chemical. It's kind of you hit it on the head before. It's chemical warfare, right? And it's chemical warfare on very many levels, on signaling your allies, on poisoning the ground for your enemies, for preparing the way for the soldiers, right? Like, so you get the roots that are evolving some of these monoterpenes as well, and they have a job to adapt the soil, right? So to attract the good stuff that will help us benefit, to repel, right? So and it's very interesting, right? The way the whole entire kind of orchestra moves together, right? Like, it's a very well coordinated, both orchestra and ballet, right? So and then it goes a step further, because now at the leaf surface and the flower surface, you're also having these things volatilized as well, right? So you have a set of things, reactions going on underneath the ground that is based on its, in fact, influences. You have a separate program going on in a separate organism, a separate organelle, right, at the top of the plant that's responding to its environment. And these things are all beautifully, characterized. And when you look at the RNA expression in the flower and then the roots, which is, we've done this and published in some of our papers, you can see that there are different non-overlapping terpene groups that are doing their jobs on this at the same time in the plant, right? Because when you do the RNA expression, right, you break it up into different tissues, roots, stems, leaves, trichomes, whatever, and you sample the plant on the same day. So on the same day when you sample, you've got one program running here and a completely separate program running here, and they're making and coordinating production of different sets of terpenes and not interfering with each other, right? It's pretty amazing.
Mm-hmm. So I think we've covered why the plant makes these things. Let's discuss now what exactly are cannabinoids and terpenes. How are they similar? How are they different?
So cannabinoids and terpenes all start from the same building block, right? So they're all isoprene based, right? And they all share common precursors, right? So specifically one precursor for terpenes, right? So GPP is the common element, and it's made in plastids. Is it the MBEP pathway or the other? I forget. Some of those details. I gotta look at the pictures, right? So, but there's a plastid pathway that turns isoprene into GPP, right? GPP is then shuttled out of plastids into, or either kept in the plastid, right, or shuttled out of the plastid into the cytosol, and you have different terpene sets and different, that are then expressed based on whether an enzyme can find GPP or FPP as its precursor in the reaction, right? GPP is also shared by the cannabinoid system, right? So you make GPP and then it's shunted into trichomes because there are, the trichome is a cell. It's an extension of a cell group, right? You get a base formed around the, where the trichome is about to elongate. You get, some thickening of the wall, you get some stuff, elongation, and then you get the glandular top. And then at the base of the gland, at the top, you have these secretory cells, which are essentially kicking the precursors into the glandular trichome head, which is where stuff is happening, right? So, okay. And there's evidence that it happens along the shaft of the trichome as well and stuff like that, right? So, but at the end of the day, right, what we're doing is we're making a pool of this precursor GPP, and depending on what either compartment or it's in, or what enzymes it's exposed to, it's going to either make things like beta-myrcene, right? GPP goes into a number of the terpene pathways, and beta-myrcene is one of the GPP. Most monoterpenes come from GPP, basically, right? So, and then that pool is shared, right? Now, this is an important consideration, right? Because GPP is the common precursor for both terpenes and cannabinoids, right? This chase of THC, right, is in fact limiting the terpene production, right? Because it's a shared pool, right? So you make a bunch of GPP and then it's either turned into cannabinoids or terpenes, right? So by forcing one direction, right, and this is another typical thing that people don't think about in terms of cellular biology, right? It's carbon flux, right? Like literally you have energy coming into the cell, these precursors are made, right? And when you have a pathway that you have to fork one precursor, you can never make a lot of both.
I see.
One has to suffer.
So, I guess the lesson here is cannabinoids, including THC and terpenes, all of the things that you can smell when you smell a cannabis flower, they share some common ingredients.
Yep.
So biochemically, they share some common ingredients. Those things can kind of get shuttled one way or the other biochemically in the plant. And there's going to be some trade-offs in terms of which cannabinoids and terpenes are actually found in any particular plant. Absolutely.
Right. And the higher the THC, the lower overall terpenes on top of it, right? So, and this gets into, unfortunately, we've been chasing THC so long, right? But recent studies have shown that 28% THC doesn't get you higher than 22 or 25% THC, right? So that it was a single study that came out of CU Boulder, but it's still, right? So there's a study that shows that from the effect from the experience side, people who were given either whatever the numbers were, one was 28, one was lower, they could not tell any difference, right? So, and then this harkens back to, for those of us of the older generation, right? Like we were getting very high on 12, 14% THC Panama Red and Acapulco Gold, But at the time, those things, right, those things were known for their bouquet and for, assuming you weren't getting Mexican brickweed, right? So if you were getting good, cannabis that was coming across the border, these things were known. They had their, they had their mystique about them because they were so far different, right? So, and these were not testing high. Like we have data from it. We have DEA data that goes back and shows that these things were not testing at 25%, right? So it is possible to get high, very high, on lower percent THC and the right terpene combination, right? So this has been the big fallacy that's been, unfortunately, pushed on the industry. So that you got to have THC.
What are so today, modern commercial cannabis in the U.S. today? What are the typical THC content? What are the THC percentages that we tend to see today? And are there limits to that?
That's a great question, right? So one would think that unless you bolster overall biochemistry in the plant, it would be, I don't think people are going to get 45, 50% because the plant has to have enough material to do other stuff like grow, right? So I think realistically, getting a lot more cannabinoid out of the plant without optimizing the genetics, like really going through and doing what we've done in ag tech. Let's say, we look at corn, right, where corn yield and sugar content and those things have been going up. There was an explosion of it, right, when we first got into molecular biology and sequencing and all that stuff. And then as we get further, there's been less of a drastic climb, but there's still every year there's a few percent increase in yield or content or whatever, right? So because of the optimization of the genetics and the breeding for better varieties and for better combinations, right? So I think we can do a lot of that without GMO, right? And get to a point where we do have a plant that is built better from the ground up. And so, there's a lot of basic agricultural genetics that we still have yet to unleash on this plant, right? That we haven't done it. There's no research, when we think about corn, right? Like the corn genetics program, right? Like for us to get where we were, it took $300 million and like 50 labs or 40 labs across the United States, right? So it was a huge input to get to where we are, right? So that's not been done in cannabis, right? So, and I think once we do some of that stuff, because we have built the tools already in corn and wheat and all these other things, we know how to do the sequencing and the bioinformatics now. It's not new science. I think, a fraction of that investment, like $100 million, with the serious academic labs and and industrial labs, working hard on it. I think we turn this plan into something that we're surprised about.
So why is it today that in commercial cannabis in the U.S. and basically everywhere in the world today, you see so much THC in these plants and you see so little of these other minor cannabinoids? What are some of those other minor cannabinoids and why are they so minor?
That's a great question. So, and this is most likely the result of intense inbreeding without good science and genetics to understand what we were dealing with, right? So, and so we can see some of that evidence by looking at CBD varieties and the minor cannabinoids that are available there, and THC varieties and the minor cannabinoids that are available, in the THC varieties, right? Dominant varieties. So, and this is a paper that came out of Canada. I forget whose lab it was. Might have been Jonathan Page's or some, associate of his. But what they showed is that when you look at the chemistry of the plant, right? So you see a lot more minor cannabinoids of different types in CBD lineages than you do in THC lineages. And that's not surprising, right? Because when you have intense breeding for a specific thing, right, you tend to lose non-important or things that are not under the selective pressure for that particular thing, right? Unless they are in fact, right, linked by a genetic physical, association, right? So if you're breeding for THC, things that are in linkage disequilibrium around THC are going to come along with it. But what you'll see is that you'll see, the alleles that you want for THC will be bottlenecked, but you'll see like CBC genes. There's a lot of variation in CBC genes, right? So because nobody's breeding for them, right? So there's no selective pressure on them, but for THC. But things that are not only good, things that are further away down the chromosome, instead of, people tout 1 centimorgan as this kind of magic number. If it's within 1 centimorgan, you tend to, it's hard to get, I think it depends on the size of the genome and the plant, right? So, and chromatin structure, right? But at the same time, right, things that are 5 or 10 centimorgans away, right? Those are easily recombination, so you get recombination. So in those situations, you could see where things further away from the thing that you're looking at, THC, or the thing you're selecting for, right? And again, this was all done by seat of the pants, right? Because we didn't have genetic tests and we weren't doing lab tests, right? So it was basically, did this get me higher than I think I got last year, right? That's basically, right? And so under those conditions, without a lot of oversight and understanding of the genetics, you're going to lose stuff. And we've seen it in a lot of organisms. If you look at the difference between teosinte and corn and maize today, huge difference, right? Let me take that back. So 4 genes or so that caused the difference in the shape or whatever, but the basic genomes between the two, right, are really quite different now from the ancestor to what we call maize now.
So basically what you're saying is, because people were cultivating and breeding cannabis illicitly for so many years in the dark, literally, they weren't doing rigorous scientific testing. They were just sort of paying attention to one output, which is, how psychoactive is this for me this year compared to last year? And that caused selection for just production of THC, and you tended to lose all of the other things that are there. So does that mean, in effect, that despite the wonderful diversity that you actually see in commercial cannabis today, that's still only the tip of the iceberg? There's actually a lot more diversity that could be there that actually has been lost in some ways.
Yeah. So I don't want to steal one of my scientists' thunder. So we're getting ready to submit a paper, but we just recently kind of looked at terpene diversity across a number of genomes, right? And because of Frontrange is really well positioned. We have our European division which is a collaboration with Crag IRTA, and so I have a whole research arm there. We do a lot of genomics there, and so what we've been able to do, and even looking at some other cultivars, right, that have been sequenced and their genomes published in the United States, right? So through various publications other than ours, you can see that there's a really big difference in some of the structural components of the chromosomes when you look across different genetic backgrounds, right? Like, to the point where, and to me, this is really, satisfying, because I've been saying for the longest time, I can't remember the first time I mentioned it was some seminar I gave. It might've been at MJ Biz, who knows back when, but I said, when we are able to do haplotypes and haplogrouping like we have done for the Human Genome Project, right, where we can look at what segments of DNA were carried ancestrally because they're in the largest proportion of the population, and then you can see the segmentation of the genome regionally. You can see haplogroups that are regional, haplotypes that are bloodline, right? So when we get to the power and the coverage of the DNA, right, sequences from around the world, we're going to see almost identical things, right, in haplogroups and crossovers, because humans when they left where they were, they took their hemp and went to other places. And if there was hemp there, that hemp interbred, right? So just like when humans went from point A to point B, they may have at some stopped interbreeding amongst their population and started outbreeding as well, right? So this is why, and so, so what we are in the midst of building, right, is this understanding of what parts of the genome carry what bits of information that are now in the hybrids we see today, and how does that affect them, right? Not all terpenes came with all plants. Some things from Southeast India or Southeast Asia had a particular group of terpenes because of their environmental needs, right? some other ones from, Europe or from South America had theirs. And it wasn't until you saw these populations end up in the same place and interbreed that we got some of the diversity and some of the things that we're seeing, right? And so that's the beautiful thing about cannabis, is that, even with the loss of some things, as long as we continue to go back and outcross to more ancestral, I won't say land race, right? I will say more ancestral populations that are, feral cannabis for all intents and purposes, right? So we will find these things, and that is evidenced by the fact that we find a lot of minor cannabinoids and other groups, right? So we're talking about cannabinoids and terpenes here, but we find not only other minor cannabinoids and interesting terpene diversity in things like hemp and grain, fiber hemp and seed hemp, right? But we also find some of these interesting minors on the CBD side as well, with things that literally, unless you have the ability to have a standard for all 148 cannabinoids, you will not be able to determine what this is. Again, and I have population genetics we've been saving that have a number of these things, and it's time, without NMR and purification to 100%, and NMR, we will never identify what these compounds are, other than we know it's an acid cannabinoid because of its structure, right?
So the same way that we have had such a focus and an overabundance of THC compared to these other minor cannabinoids, I suspect that we've also had some particular terpenes reach high prominence in commercial cannabis at the expense of others. What are some of the most abundant terpenes in cannabis today, and what do we know about them?
So you're absolutely right. So beta-myrcene and beta-caryophyllene end up to be the two of the top, right? And again, right, largely those two things, end up being, I think it's some ridiculous.
It's like 2/3 or 3/4, I think.
Yeah, right. Yeah. Just those two, right? So and so that's an example of. I'm glad you asked that question, man. So so that is an example of exactly the bottlenecking, right? Like, and again, it comes from predominantly, right, because a few varieties and what they carried with them, because they were high THC producers. We kept hitting high THC producers together, and and that's what I was talking about. So if you look at U.S. varieties versus European varieties, you can see there's really significant differences in the bottlenecking that occurred in each region, right? So, so now, right, I think, one of the other interesting observations is when you do outcrosses, right? So the outcrosses are also now kind of, controlled by whether, I can't talk too much about it, but when you look at certain varieties that have chromosome deletions, right? Like, and so now you have things that are not producing certain cannabinoids because that piece of the chromosome is missing, right? So trying to outcross those things, right, creates all sorts of havoc. Right. So now you get to a point where we've done some damage in some ultra specific varieties to the point where it causes like an imbalance, and you have a very unhappy plants when you put them back together, and the offspring are really weird. Hermy, grow poorly. Right. So I think, wow, I think I just totally lost my train of thought there.
Yeah. No, well, why don't we just bring it back to some of the more common terpenes? So there's a handful that are really common. Right. What do we know about, if anything, what they actually do physiologically?
Okay. So yeah, I'm sorry. So the other ones that are in that upper group, you see alpha pinene, you see, so we talk about myrcene, beta caryophyllene, alpha pinene, you see limonene, right? And to a lesser extent, I think you see some stuff like beta farnesene, and there's one more. There are 6 really predominant ones, and then there are a few more that are 2 of the predominant ones kind of coexisting together, right? Like, alpha pinene and myrcene sometimes, or beta myrcene and limonene, so, and then, so those are realistically the majority of the 70%, 75% we were just talking about come from combinations of just those 6, right? Like the ones that we just talked about, right? So, and then, terpene, when you do extended terpene profile, when you look at every terpene, when you invest in that kind of science, right? So, what you'll see generally is that you'll have a first group, which is produced at about 5 to 10 times more than the second group. And then those are then produced at about that same level over a third group, right? And in that third group, you have really esoteric stuff like, guaiol, neuroladol, that kind of stuff, right? So then in that second group, you have some of the more common things, right? Like the beta ocimines and the beta pinenes and some of the other stuff, right? So now what's interesting, right, about those is that, just like the cannabinoids, we're starting to see some losses there, some things that travel together there. And if when you look at the industry historically, not even maybe historically, but I found an article that was produced by SC Labs after the last, the High Times Cup or whatever it was they did, right? And they talked about a paucity of varieties and 3 terpene categories, right? So I think, again, there's an example, just like with the cannabinoids, where we have focused so much because of the sense of smell and bag appeal, right? So the 2 things we go after in the industry are how much THC and what does it smell like, right? So, so we're starting to see some losses there. Now, why is that important? It gets to the point that you were segueing to is the physiological effects. Well, a lot of these terpenes are, in fact, the mid set is as important, if not more important, than the big set, right? And by that I mean, like, so things like, linalool, right? It takes 10 times, or maybe somewhere between 10 and 100 times less linalool to smell than it does beta myrcene, right? So there are times where, we have to not think about the plant from what's the most abundant terpene, but what is the combination of terpenes, because you can have a less abundant terpene have a stronger effect than the most abundant terpene in the overall effect profile, right? So now let's get into the effect profile. So effect profiles are a thing, right? And some of it comes from the fact that we have to understand that the plant is not doing this for us, it's doing it for itself. And so the first rule is plants are not confused, right? So when they are making ACH inhibitors, right, which for us cause a higher cognitive function, and let's say alpha pinenes and those kind of things, right? What those things are doing are actually a completely different thing, where it's there to attract some organism that's going to parasitize the thing that's eating it, right? So, or it's going to, or if it's making, let's say, linalool, right, or neuroladol or guaiol, it's there to paralyze that insect from contact inhibition, right? So, and those things tend to be alcohols that are sedative to us, right? So there you can see kind of, a correspondence, right? It's a contact inhibitor, paralyzes it, makes it lethargic and paralyzes it, doesn't move, so it's easily foraged by something else, right? And yeah, we get sedated, we like it's relaxing, right? So if you see a predominantly elevated terpene, the predominant terpene is an uplifting terpene, you will not see a bunch of sedative terpenes in that plant, right? It's because the plant, by and large, is not confused, right? Even though we've mishmashed a bunch of hybrids together, because it's still responsive to a biological program that's being triggered by the environment, it's still producing in a coordinate fashion the things that it needs to survive, right? So once we get to that point, we can now start to understand now how looking at the various complete terpenes gives you a different picture than, oh, this is beta myrcene, right? And beta-myrcene is this much maligned thing because beta-myrcene, by and large, produces a mildly, at best, sedative effect, right? But yet it is the one, because it's in indicas, right, that always gets hammered as being the sedative terpene. But it's not, because if you pair beta-myrcene with alpha-pinene, you get an uplifting, relaxing effect. If you pair beta-myrcene with linalool and or limonene, or both, you get a very sedative effect, right? So, and we know these things because for generations, like 150 years, we've been studying terpenes and essential oils by GCMS. We have a very standardized way to fragment these things and to understand their fragmentation patterns. And then we can go and we can take a look at these things, identify them with 100% accuracy, and then say in mouse models or in aromatherapy trials that have been done on humans, what is the predominant effect, right? And we've even gone further where we've actually put, either patch clamped mice, or we've put, EEG, on receivers on people's heads, and fed them alpha-pinene and measured their brainwaves and shown that, look, alpha-pinene gives you a very focused alpha wave. Beta-pinene raises the beta wave, right? So and so these things are known physiologically, both in animal mammal models, which is sometimes the best we can get, and in some cases they are directly on human studies, right, through aromatherapy and observation, right? So so once you start to look at the terpenes as an integrated set, not just what's the highest or a single one, you get a different picture of cannabis, right? And that picture of cannabis is, in fact, one that is highly medicinal and therapeutic if you pay attention to what's going on. Further, right, you get these synergies between the cannabinoids and the terpenes. And so that concert in our physiology causes us to respond in a certain way, right, through our endocannabinoid system, right? But there is actually a lot of logical, kind of correlations between what the chemistry is, what the plant's trying to do, and then what the effect in the human is, knowing that the plant didn't evolve these for us, but for its own survival.
So let me try and summarize some of that. Basically, what you're saying is different types of plants with different genomes living in different environments are going to want to produce different cocktails of chemicals to protect themselves and to thrive in their environment. So, for example, there might be one plant with one set of genes living in one environment, and it has to worry about certain types of insects coming around and eating it. And perhaps the best way to deal with those insects is to secrete a chemical cocktail that makes those insects lethargic and sleepy so that they get picked off by other critters, and thereby the plant is protecting itself. But maybe there's another plant with another genome in another environment, and it really has to worry about other insects or maybe some other type of mold because it lives in a warmer climate. And so it's going to tend to make a different cocktail of terpenes and other things. And it just so happens that when those two distinct cocktails of terpenes and other compounds get made by two different plants in two different environments, that when a human goes and consumes one plant compared to the other, that's inevitably going to have some kind of different physiological effect because that cocktail is different between those two plants.
Very well said. Boy, I wish I had said it that easily.
Well, so we haven't said it explicitly, but this gets to the idea that a lot of people talk about in the industry of the entourage effect. So can you just sort of restate, you're sort of restating what you were saying in some parts a moment ago, but what is the entourage effect, and what are there clearly different entourages that we see in different types of cannabis cultivars?
So let's start with the second question. And the answer to that is absolutely yes, right? We do see different entourages in different cannabis cultivars, for sure. Also, you can also sometimes change a single cultivar's, entourage profile based on growing conditions. That gets back to what we were talking about before, that, phenotype is the equal to genetics plus environment, right? So now, the entourage effect essentially is, in fact, the combination or the overall result, right, from the combination of that chemical cocktail, right? So, and that chemical cocktail is not just the terpenes, but it's the terpenes and it's the cannabinoids, right? And these things are known to act synergistically. If you look at some of the, the properties of both cannabinoids and terpenes, right? So many of them are antiviral, many of them are antifungal or antibacterial, or anti-inflammatory, right? So THCA, a great topical anti-inflammatory. Well, some of the terpenes themselves are also anti-inflammatory, right? So when you have, and further, right? One of the most important things about, I think, the entourage effect is the ability for the terpenes to help predispose the body for the actual cannabinoids to take effect, right? And I think that's an important consideration, right? So you have things like beta-ocimene, and you have transmineralols. So some of these things are outstanding skin penetrants, right? So, and so that means they get across, lipid membranes. So if you have in the packet that you're carrying in the smoke or in the edible or whatever, or in the topical, these things that already help smooth the way through, right? You get a much better effect in that combination, hence the entourage. And you get also, on top of that, the entourage can also sometimes cause synergistic reactions because the combination is greater than the sum of the parts, right? So because you have them interacting and they are, many of them are doing the same thing. So instead of having one, right, you have three that are acting through similar or overlapping pathways to cause anti-inflammation, right? So, and this gets to, if you want to get really deep into the biochemistry, this is pathway signaling, GPR protein triggering, and all this other stuff, right? So, but to not geek out totally on that, right? What we see, right, is that, because of what we define as these chemical cocktails, right? So now different cocktails are going to produce different effects, right? So is the cocktail predominantly one that's going to cause lethargy or sedation? Or is it one that's going to cause, ACH inhibition, which is focusing, energetic, uplifting, right? So I think, and further, right? So if you take a look at chemical profiles, right, and this gets into, chemical classification and schema, right? Like, how do you go about trying to make sense of the broad diversity of terpene profiles that we see in the cannabis plant, right? So if you start by what's the most abundant terpene, right, and to get some sort of hierarchy, and then and that's where you get largely the 6 or 7 groups based on those first most abundant ones, right? And then you then create a hierarchy in each group or each clade, right? You end up with some very interesting separations, right? So you can have some that, that might be in the energetic, but yet causing pain relief. Some might be sedative and cause pain relief. Some might be sedative and anxiolytic, right? But not pain relief, right? So how do we get to that point from this discussion of terpenes and entourage, right? Well, when you dig down deep enough, we know things like alpha-pinene is an anxiolytic. It helps reduce anxiety, right? We know things like limonene also, right? Another one, right? So, but they're operating from, one's focusing, right? One's relaxing. So they're approaching it from different parts of the body, right? And so when you look at a real deep dive into a chemical profile, you can actually see, and you go back and you do the research that we had to do to say, okay, what's every known medical response caused by each of these terpenes is how we started that path, right? Mm-hmm. So what is known medically about these things? And you put them together, and you look at the profiles, suddenly now it makes a lot of sense because you can see these minor things like, transneuralidol, guaiol, which are tertiary substituted alcohols, right? And in cannabis, these tertiary substituted alcohols all cause sedation, right? So there's a certain logic and there's a certain kind of pattern that falls out of it when you start to look at entourage seriously.
So what you're saying is there are, in fact, distinct combinations of cannabinoids and terpenes that tend to show up in different types of cannabis cultivars. And if you categorize or you group those different cultivars based on their chemistry, you can see that these different combinations are present in each one. And when you start to think about, what we know, whether it's very much or just a little bit about what the chemicals present in each one actually do, you can start to make some educated guesses, at least, as to what these different kinds of cultivars will actually do when a person ingests them. So I guess my next question for you is, when you take, given that we're talking about cocktails of drugs here, whether or not you're talking about the psychoactive effects or the medicinal effects, at the end of the day, these things are going to be driven by the drug cocktail one is consuming. So someone like you looks at the phytochemistry and starts to group the different types of plants based on their chemistry. You see that there are these distinct groups. How do these groupings that are based on chemistry relate to the groupings that one is actually going to encounter in a legal cannabis dispensary today? And so, to help get to that question, based on all the research you've done and all of the chemical categorization work that you're aware of, if you go into a dispensary today and you talk to a bud tender, the person in the dispensary that's going to be recommending a product to you, they're going to say things like, well, we've got indicas over here, they'll make you relaxing. We've got sativas over here, they'll make you energizing. Is there any clean relationship between what you'll be told in the commercial setting like that and what you're talking about based on your chemistry research?
So, that's a complicated question. But so the answer to that is as with all things cannabis, yes and no, right? So, and some of this goes back to earlier, we were talking about form and function. So in the old days, sativas carried one typical chemical cocktail because of where they evolved. Indicas typically had theirs because of where they evolved. Humans came together and screwed it all up, right? So now we have plants that are indica looking, but sativa profiles, or sativa looking with indica profiles. However, when you look at some of the genes that are responsible for that, right, there was at least at one point, and there are certain terpene genes that seem to be indicators of sativa-like performance. And the reason why I say that is because when we did a lot of this bioinformatic analysis of iso-seq data, RNA-seq data, right, which is from trichomes and expression, right? Then we took that a step further, and I had my team actually clone these things into bacteria, put them in a test tube, feed them precursor, measure what was made, and then put it on a chromatograph, right? So on HPLC. And what we found was there are, in fact, some terpene genes that do make the compounds necessary to be a sativa, right? And by that, when you look at traditional terpene cocktails from sativas, you had things like alpha-phalandrine, 3-carine. There were some very specific terpenes that contributed to that energetic, uplifting feel that sativas traditionally gave you. Well, lo and behold, there are some terpene genes that when you give them FPP and GPP, right, they come out with a combination of terpenes that it looks, in fact, a lot like what you would see in a sativa, the alpha-phalandrines.
Can you just clearly state what that combination is?
Hold on a second here. I got to pull up some data here. Where is that? I'm looking for a particular figure that I can read them off of. Because I have one of my one of my scientists is getting ready to give a talk about it. Give me 1 second. I know that I'm wasting a bunch of time here, but I don't have them memorized, and I need to get it off of the actual figure. Let's see.
I see, but you think that there is such there are genes that correspond to a plant that will produce such compounds.
Yes. So, in fact, it's largely the presence or absence of one specific gene, right? Like so, and one specific gene, actually, I mean, it was we were all like, I mean, I don't want to use bad language, but like the entire team after this, my team member presented it, we were like all like, holy. Like that's the sativa gene. Like that was literally our response. It was every terpene that had been traditionally associated with a sativa effect came out of this one gene. And many varieties do not have this gene, right? And you find this particular gene in some very, traditional sativa-like cultivars, right? So,
and what are those?
Things, most of the equatorial, plants that are the taller, lankier ones still that you find the equatorials. Things like super silver haze is one of the old school ones. Red Congolese, which is Red Congo, which comes from Africa. A lot of the African quote unquote land races are indicative of that sativa lineage.
Yeah. So some of these, I think, including Durban poison, I think, including super silver haze, in my research, they have been associated with a terpene called terpinolene. Is that what you've seen?
So interesting. Terpinolene you can find both in sativas and non-sativas, right? Terpinolene gene. So many genes make terpinolene. However, one of them makes terpinolene in conjunction with these other terpenes, right? So in the gene that I'm speaking of specifically, and I'm not sure I should give it out just yet, but it's going to be on our next publication. So, but this particular gene, it actually does, its profile, terpinolene is in fact one of the things that comes out with the 3-carine and alpha-phalandrine and some of these other ones. So.
Interesting. So do you, taking the consumer standpoint for a moment, if you're a cannabis consumer and you walk into a legal cannabis dispensary today, are the labels given to different cannabis products, are the labels Indica and Sativa going to actually tell you very much about the effects in that dispensary today? Generally, no.
No, because largely those decisions are being made based on the what it looks like and color, right? There's nobody doing deep enough dives into the chemistry. I take it back. So there are a couple labs, right? And I don't want to do any product placements for anybody, but there's a couple labs in California and around the United States that are doing extended terpene profiles and are doing them well, right? So but unless there's a COA and unless everybody or every product from that dispensary shelf is coming from that lab, there's probably not enough information for the general populace to make that distinction. And it has to be an extended terpene profile. The top 4 or 5 terpenes, top 6 terpenes does not give you the information needed to be able to make a determination as to whether or not you have a sativa or not.
I see. So what you're saying is that based on all the complexity that you mentioned to do with all of this stuff, all of the terpenes, all of the underlying genetics, how everything's been hybridized and mixed up over time, that the way things are named and labeled and organized in the legal cannabis shops today isn't actually going to be very informative to what you're going to get in terms of the effects.
Yeah, and I can't argue that. That's 100% correct. That's been my position for quite some time, and it's made me rather unpopular sometimes. So, but yes, it is the truth, because what it means is the end to brands, right? And I hate to say that, right? But like realistically, Gorilla Glue or whatever you want to call it, now, GG4, whatever, right? So if you grow it 4 or 5 different ways, you're going to get different chemobars, right? You're going to see differences in that chemical profile, right? So again, right? Unless you have a more extensive chemical profile, right, and you run that chemical profile through some sort of algorithm that says, based on what you have, this is what you would expect, and this is where it should be classified as an indica, sativa, or hybrid, right? I don't think you don't have enough information coming out of most labs and into most dispensaries to make those calls.
Why is it? So given the lack of correspondence between the way things are labeled in the store and the way they're marketed and the actual chemistry and the empirical reality of what we're talking about, why is it that, when you walk into a dispensary, people are so confident when they say, this indica over here will make you sleepy and this sativa will make you energizing? Is it all just, marketing posturing or what?
So some of it, I think, is favorites, right? So bud tenders, know what they know because they are in fact themselves, consumers, right? So, and that's in fact some of the problem, right? So a bud tender who loves to be couch locked and his mind free, right? That may be a very annoying kind of experience for somebody who's trying to clean their house, and suddenly finds themselves unable to focus on anything because they got really high and, on an indica, right? So quote unquote indica, right? So a lot of it is the industry, well, THC, right? And then after that, it is favorites and or brands, right? And I think that's largely what happens. I think you do have some bud tenders who are starting to understand, and some older bud tenders who have been around a while understand the importance of the entourage or the terroir or whatever you want to call it, right? So, and I think those people are the better bud tenders, right? Because they will talk to a person longer and say, hey, what is it that you're dealing with? I mean, they have taken that next step because they understand, hey, well, maybe you don't need this if you have anxiety. We need to get you away from that to something like this, right? So, but largely, and I stopped going to dispensaries a couple of years ago, right? Because I grow in my backyard. It's legal in California. I grow in my backyard, right? So, but when I was going to dispensaries, right? So my observation, and even at some better dispensaries, right, some top-notch dispensaries, was that the education of the bud tenders was not always consumer to the job they were supposed to be doing, right? Like commensurate, right? Sorry, right? So here they were trying to be a purveyor of information so people can make intelligent decisions, and instead, what they were like, oh, dude, this one got me so high when I tried it, right? So it depends on what the consumer wants, right? But I think part of what we are talking about here is also the endocannabinoid system, right? So, and I meant to, make this connection before, is the bud tender who is not talking to you about what you want and trying to show you terpene profiles to affect what you want, and is trying to sell you based on THC percentage or what their experience was, is largely misleading you, because if you're not built just like them, you may have a crappy experience, no matter how good experience they had.
So do you think it's possible to, is there a viable resolution to this problem where, is it going to be somehow possible for us to bring the marketing claims that you see for cannabis products in alignment with the empirical reality of what is actually inside these things? Is there a way to do that in your view?
Yes, I do. I think there is, but it requires, science, right? So it does require that people kind of standardize testing, and then when we kind of share information, right, and that we are able to come up with an understanding of um, what constitutes a a set of lab tests that can be used for deeper investigation into the potential of the plant, right? And that's kind of I was trying to formulate how I want to say that as I said it, but what we're trying to get to is an understanding of more terpenes, right? So 6, 8 terpenes. What most regulatory bodies require is not helping us, right? And the 5 cannabinoids is not helping us, right? So we don't get any additional information that may in fact impact, right? Like, if you're not measuring THCV and you have a plant that has a little bit of THCV, depending on how much THCV, that might change the spike, like how fast it hits you, how hard it hits you, but you don't know that because you haven't seen it, right? In the COA, right? So, and the same thing goes with the terpenes, right? So in order for us to be able to understand how the terpenes are going to predispose the overall effect of that flower is, you have to have a certain number of terpenes measured to a certain precision, right? And in some cases, isoform distinguishing is going to be important for some of these things because some of them have many isoforms, and not all isoforms behave the same way, right? So alpha pinene and beta pinene behave differently. S-limonene, R-limonene behave differently. Believe it or not, S-linalool, R-linalool, right? One of them is a sedative, and one of them is an energent, right? So is actually makes you kind of like racy, right? So we have to know the type and the form, and then from there we have to build systems that allow us to say, okay, based on this chemical profile, this is the effect you'll get from this, regardless of the name, right? And because Blue Dream from any given grow could fall into 2 or 3 different subclasses underneath its major class, right? And so we need to get to the point where we treat it like a medicine, we analyze it like a medicine, and then we present it like a medicine. If you're looking for something that will, in fact, help you with anxiety or relieve pain, these are the choices you should look at, not these. So,
given what we were talking about earlier in terms of breeding different cultivars and comparing cannabis to other things like corn, where we've spent a lot more time and a lot more money so far on those breeding programs, how close are we to being able to breed or create genetically modified cannabis cultivars that have arbitrary cannabinoid and terpene profiles? Is that something that's a couple of years away, or is that a decade away?
So I believe, right? So there are already knockouts. I'm not sure that anybody has actually perfected the, adding not, genes that are not present yet. Some of it requires stable, some of the techniques that we just got, like, being able to stably transform cannabis, being able to regenerate from single cells or callus. Some of that stuff, technologies just came on board, right? So some of that needs to be developed more. I know people have been working with, CRISPR and have set the stage, right? So even my team has created CRISPR guide RNAs and stuff like that, just for some of the genomics that we have built, so that if we need to go to a next stage, it's there ready, right? So, but the reality is that, we are not far enough along in our understanding of the genetic diversity and or the chemical diversity, right? So, all of this data that we're talking about is based on very limited access, right? Like, it's not like we've gone everywhere and we've tested we chemically flowered out every type of cannabis around the world and seen what we've got to work with, right? So there's still a lot of unknowns there that we have to tap into that allows regular, traditional breeding with good science base, right? So, and development of genetic markers for marker assisted breeding and that kind of stuff, right? So there's a whole bunch of stuff that we haven't done yet. But I think to get to it's probably only a few years off, like 3-5 years off before we probably could produce regular genetically modified organisms that have arbitrary terpene profiles. As of now, right, because of the marker studies that we've done, we can already do traditional breeding, right, to get unique combinations. And my team's been working on that based on our studies, right? So we've actually started to breed, plants that have unusual terpene profiles specifically because they have unusual terpene profiles that are not like everybody else's, right? But because of the limitations of these not being genetically modified organisms, right? It's back to the traditional breeding, right? So you did the cross, now we have to pheno hunt and screen with the markers and pull out the ones that we want, and more breeding, and it's an iterative process, right? So, but I think we are already at the point where we can breed for unique terpene profiles, right? Because of markers and traditional breeding and understanding which plants have what. 3 to 5 years, no more than 10 for sure, but I think probably 3 to 5 years, people will have genetically modified cannabis out there ready for sale. Whether or not they'll get into the market is a different story, right? Because remember, there's a whole history of getting genetically modified organisms into the market of any kind, right, produce or whatever, in the United States, right? So just because they can do it doesn't necessarily mean it'll be out of the market anytime soon.
Mm-hmm. Are there any if you could snap your fingers and just make an arbitrary plant with an arbitrary cannabinoid and terpene profile, what would that look like? I guess what I'm asking is, are there any cannabinoid and terpene combinations that you're particularly intrigued by because you think they might have interesting effects?
I'm agnostic to that. So for me personally, right, I'm a heavy sativa, uplifting, kind of jump out of my skin guy. So all of my breeding reflects, or my early breeding, now that it's no longer just for me, it's for the company. I tend to look at things in a different way, but all of the early varieties that we released at Frontrange were from stuff that I was breeding for me. And I'm a heavy jump out of my skin, high edge. So I was breeding for things like that had high energy terpene profiles and THCV, if I could get it in, right? So, or that kind of stuff. So I'm completely abnormal. Most people don't like to feel like they're jumping out of their skin. I find that my comfort zone, right? So, if I could snap my fingers together, I would have a beautiful, so in my experience, the varieties that I've created that have THCV don't behave well. So they're problematic. They're weird. They're weird plants, right? So high THC plants. So if I could snap my fingers together, it would be the magical plant that had beautiful flower structure, behaved well, and had, very sativa-like terpene profile. That would be, that'd be mine, right? And I'm pretty sure that would be pretty popular because something like that would be, make you appetite suppressant. It would be mind focusing. It would be get shit done weed, right? So.
All right. Well, Reggie Gaudino, thank you for your time. Are there any final thoughts on the general topic of cannabis and commercial cannabis chemistry that you want to leave people with?
Yeah. And it's something that I wish people paid more attention to. So science is your friend, right? Like I know, so science costs money, and I know it's been a bad word in a lot of the regulated industries, right? But science is your friend, right? So not just talking about, contaminants and quality assurance for safety, right? We're talking about, knowing what you're putting in your body and being able to use the cannabis plant effectively. The better you know the plant and the better you know your ailments, the more, the sooner personalized medicine through cannabis can become a reality for people, right? So there are so many compounds, right? And you and I talked about cannabinoids and terpenes today, right? So, but think about all the compounds we didn't talk about: flavonoids, isoflavonoids, carotenoids, which we already know are part of the superfood family, right? Antioxidants, all of these things exist in a fair bit in cannabis, right? So there are, and one last thing, there's so much unknown production in trichomes. One thing I didn't talk about is when we do all these RNA studies is that we find, a third of the production of capacity of the trichomes is not even cannabinoids or terpenes. We find these other genes that are highly expressed at RNA. They're part of the berberine breach family, but we don't know what they make. right? So it's a related compound. It might use a similar precursor, but there's a whole other set of chemistry in trichomes we don't even talk about, right? So chemistry is our friend, and the more we know, the more we know. So,
all right, Reggie, thanks for your time.
Thanks.